This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1219, recorded on the 16th of May, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Howdy there, Vincent. It's great to be here. Just to preview, just the two of us, right?
It's just the two of us.
This
That's
is
right.
cool. This is we can do this. We've done this before. We've done it before with thousands of miles between us. So that's cool.
That's right. It's 23C here. I'm in New Jersey today because the commuter rail, New Jersey Transit, the engineers went on strike. So all the trains are shut down. So fortunately, I have a studio here, as you can see. I like the incubator better, but at least I'm here.
So what we got here is, let me make sure I'm looking at the right thing here. Yep, we have 96 degrees. And so we have been experiencing since Tuesday. This is Friday. So Tuesday? Wednesday. I don't know. What they were calling a heat wave. It was nice and cool and wonderful, leading up to this. And then there was a 40-degree increase in, in one day. It went from like in the morning 60 degrees to the afternoon 100 degrees. And that was the beginning of a heat wave that was 101 one day and then, a pleasant 99 the next day. They called it a heat wave, but if I look out the next foreseeable future, that is 2 weeks, we're talking mid to high 90s. So this is more than a heat wave. I think it's summertime. They may get a few more days of less than 90, but not in the foreseeable future. Here we are, Austin, Texas.
I already told you, if you like these programs, can't tell you too many times.
No.
We'd love your support. We talk about real science here, not lies, not misinformation. We don't do ads, so we depend on listeners to support us, and so far it's worked. So please help us continue. Microbe.tv/contribute. ASV 2025 is going to be hosted by McGill University, and it will be held in Montreal at the Palais des Congrès de Montréal, July 14th through 17th. The website is asv.org/ASV2025. You can check out the program on the website, see the special events and workshops that are in addition to the normal talks. Now, deadlines. We've passed the deadline where you can register cheaper. Sorry. But registration for the meeting ends on June 30th. So if you don't register by then, it's tough. And after that, actually, the rates go up. The reservation deadline for the Campus One MTL residents has been extended to June 12th. Sign up by adding the reservation to your meeting registration. And so I have registered. I'm all set. I got a plane. I just need to get a hotel, and I'm all set.
Cool.
Looking forward to that. A little bit of news here. This first one is a little bit disturbing. It's from the FDA. FDA and CDC recommend pause in the use of IXCHIQ, I-X-C-H-I-Q. That's the Chikungunya vaccine, infectious attenuated in individuals 60 years of age and older, while post-marketing safety reports are investigated. This is not too old a vaccine, Chikungunya, an arthropod-borne virus that is not endemic in the U.S. There are some imported cases, but it's in many other places. So if you're traveling to certain places, you might want to get this vaccine. However, they've called for a pause in its use in people 60 years and older. There have been some post-marketing events like neurologic and cardiac events in people. So as of May 7th, 2025, there have been 17 serious adverse events, including 2 deaths that have been reported in people 60 through 89 who got the vaccine. Okay? Now, 80,000 doses have been distributed globally. These events may not be related to the vaccine, all right? So that is what they need to do now and figure that out. But in the meantime, since these serious events have happened in this age group, they're pausing the vaccine, which is the right thing to do. And it just shows you that the regulatory agencies react when there are problems. This vaccine, by the way, was approved in November 2023.
So I had several reactions to this. My first reaction was seeing a headline that FDA and CDC recommend pause, blah, 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 blah. And unfortunately, my first reaction is this the real FDA, CDC that I'm used to historically? Or is this FDA and CDC that are under the influence of current politics? And to what extent can I trust this report? Now, and I have to say that I just had that reaction. And that's just an unfortunate commentary on the current situation. Now, as it turns out, as you read through this, it's clear that this is the real deal, that this is FDA and CDC doing their job, okay, right, as they're supposed to do it. But if you're not, if you're not used to looking at this kind of stuff, it could be, I mean, you don't know how to evaluate it necessarily. But for the time being anyway, this is the real deal. My second reaction was that I did some math. 17 cases out of 80,000. Oh, first of all, as Vincent said, these are adverse events as reported in the database we've talked about before, which is the Vaccine Adverse Event Reporting System, or VAERS. This is the place where one can't report things that happen within a certain window of time after a vaccination that you think could be associated with the vaccine, but you don't know. You just report them, and the idea is that the agencies monitor this system, and if they see spikes of adverse effects, in particular of a particular kind, following administration of a certain vaccine, that's a motivation to look into that. And this is an example of that in action, though, as Vincent said, they've got to follow up on this. And one of the reasons that this is important is that this system is often misused by the anti-vax crowd, okay, to say, look, after this vaccine, 1,000 people died. Yeah, they may have died of heart attacks or whatever, and they may have died from, it's just that's background information, okay? So you got to know what this system consists of and how it gets used. Now, I did some math. 17 out of 80,000 cases is 0.2%, all right, which is not a small number. And assuming that's correct. And my second question was, well, is this something that would have shown up in the trials for the vaccine? Because remember, in COVID, they did depending on the product, they looked at, between 30 and 50,000 people. They were huge trials, okay? And a signal like this probably would have shown up in those trials. But if you look further into this article, the trial was in 3,500 individuals and included, importantly, a placebo. Vaccines are done in placebo-controlled trials. Everybody listening? But at 3,500 cases, a trial with 3,500 individuals, a signal of 0.2% would not necessarily have shown up. That's like less than one of these adverse effects, which emphasizes that quite often you don't see adverse effects show up until a vaccine is actually deployed and you vaccinate a large enough people so that the large enough population so that these rarer events show up, and that's why they do the post-marketing evaluation. So this is a really great, it's unfortunate, but it is the scientific process in action.
All right, one more news story. This is a bit disturbing for a different reason. I have a stat article by Isabella Cueto. Health and Human Services is going to stop recommending routine COVID shots for children and pregnant people. This is a rumor. It hasn't been announced yet, but supposedly RFK Jr. will announce this, which is disturbing because pregnant people are at risk for severe COVID, and over 200 children have died of COVID since the beginning. So why shouldn't they be protected? I don't really understand. Now, the article is interesting. It says it's not clear how Kennedy would do this since there are independent advisory committees that make the recommendations, right? So he would be going around that, which is weird. He's not supposed to do that. And so we'll see what happens. But I don't know why they're doing this. There are no...
So there's no rationale. This is new in our show notes here. I haven't seen this yet. No
I'm sorry, I
rationales
just put
presented.
it in, yeah.
It's okay, cool. No rationale presented?
No rationale presented. Of course not.
So
because, eventually they want to get rid of all COVID vaccines. That's my feeling. So this is just first step.
So all of this stuff needs to be looked at from the point of view of risk benefit. Okay. And as far as I'm concerned, if there's no coherent risk benefit analysis that says that the risk of the vaccination outweighs the benefit of the vaccination, then there's no solid rationale for doing this. Okay. Or if there's no risk from, the risk, there would have to be no risk from disease or a high risk from vaccinating. And as far as I know, neither of those applies to this situation. But there are those conspiracy-minded individuals out there who feel like there's some risk associated with COVID vaccination. And it's been tainted over time, too, because of the whole mandatory thing and all that kind of stuff. It's just completely out of whack. So I hope that this never progresses beyond a rumor, but we'll see.
So if, in fact, it happens that they don't recommend the vaccine for these groups, that means even if you want it, you can get it, of course,
Yeah.
but your insurance is not going to pay for it anymore.
Right. Okay.
So that's the consequence.
Right.
Unfortunate. And the thing that bugs me is that there's no rationale for this, right?
Well, hopefully, if it actually comes to pass, they'll have a rationale, though I can't imagine a rationale that would make any sense based on what I know.
No, I can't either. All right, on to the literature. We have a couple of interesting papers for you. First one is in Cell. Molecular basis for shifted receptor recognition by an encephalitic arbovirus. The two first authors are Xiaoyi Fan and Wanyu Li, and the last author is Jonathan Abraham from Harvard Medical School, University of Texas in Galveston, University of Kentucky, and the Broad Institute of Harvard and MIT. Now, alphaviruses are enveloped positive strand viruses, some of which can cause encephalitis in humans and in horses in particular. And some of them are called Western equine encephalitis virus, or WEAV, Eastern equine encephalitis virus, or EVE, and Venezuelan equine encephalitis virus, VIV. We have talked about this recently, but I couldn't find the TWiV episode.
We've talked about these viruses on, I think, several occasions in the past.
Yeah, but there's one more recent one where we talked about the shifting, where some of these viruses are going away, and they have an interesting word for that, which submerged. They've become submerged. And so this paper essentially tries to look at that. So weave infections, they can be mild or asymptomatic, or they can cause encephalitis, brain infection, and that can have neurological sequelae. Weave used to cause large outbreaks in North America, but they've decreased in frequency and scale. And the last case of weave in North America was 1999. And same thing happened in South America. The last case was 3 decades ago, but weave is reemerging in South America. In 2023, there were over 2,500 horse cases and over 200 human cases in 3 countries. So part of the issues in this paper are why did it disappear and why are they reemerging? Now, the virus genome encodes non-structural and structural proteins, and the two glycoproteins, E2 and E1, which are embedded in the virus membrane, this is an envelope virus, mediate binding to receptors and fusion. The weave E2E1 binds a cell receptor called protocadherin 10, okay, PCDH10. This is an adhesion molecule that happens to be in the brain, and there it regulates synapse development. The extracellular part of PCDH10 has 6 what are called cadherin repeats connected by loops that are made rigid by calcium. So weave E2, E1 binds EC1, which is the first extracellular cadherin repeat, the most membrane distal. That's where the virus is binding, right? There are other alphavirus receptors, which include a protein called matrix remodeling associated protein 8 or low-density lipoprotein receptor, LDLR. Okay, so those are the receptors, and we're going to unfortunately, there's so many abbreviations in this paper. It's very hard because you just don't remember what the abbreviations stand for. The strains isolated in North America are divided into 2 lineages, A and B, and then B is subdivided into B1, B2, and B3. And what happens is, as a lineage appears, it displaces the older ones. It's kind of like COVID variants, right? They appear and displace the other ones. Same thing with these lineages of weave. So in North America, the most recently detected one is B3, which I think I said was last detected in 1999.
I want to add a couple of things about this virus and actually check in with you, Vincent, and make sure I got this right. That is on my mind as I read these papers and will come up later. And that is about the life cycle. As you said, it's an arbovirus, right? So it's a mosquito-borne virus. And I presume that it replicates in the mosquito host as well as the mammalian host, right? Correct? I mean, there's very few of these things that are actually just mechanically transmitted. It probably replicates, I assume it replicates in the mosquito. And there'll be the normal cycle, I would think, would be back and forth between mosquitoes and horses. So a mosquito bites a horse replicates the virus, may or may not get sick. The virus is in the blood, the mosquito picks it up. The virus is in the mosquito, replicates in a mosquito, that mosquito bites another horse. Occasionally the mosquito bites the humans are infectable, can get disease, but am I correct in saying that humans are what we call a dead-end host in this case?
So are horses, actually. They're dead ends,
is that right?
Too. There's some other reservoir, could be birds or rodents.
Okay, so the actual productive cycle involves something other than either humans or horses, correct?
That's right.
Okay.
That's right.
Good.
That's right.
All right. But the whole time I'm reading this paper, I have the mosquitoes in mind, and yet they're never mentioned in the paper. We can come back to that.
Yeah, they aren't mentioned, although they're in the graphical abstract.
Right.
They're pictures of mosquitoes. Oh, actually not. There's humans and horses, birds. Birds, because I guess the birds are a reservoir. So the South America, the most recent outbreak is a lineage C. And so PCDH10 is a receptor for many weave strains, including lineage A strains, which come from the 1930s and 40s. These are highly virulent in animal models. They also bind very low density lipoprotein receptor and apolipoprotein E receptor 2. So there is a virus called Imperial 181, which we're going to talk about in this paper. This is a B3 lineage weave isolated from a mosquito pool in California in 2005. It's not pathogenic in animal models. It does not bind human or mouse PCDH10, but it does bind PCDH10 of the house sparrow. And so those animals are presumably a reservoir. Yes, and they say they're the enzootic host for weave.
Oh, okay, yeah, now that makes sense. I even looked up enzootic, but I didn't actually connect all the dots. Good.
Right. And also snakes. The snake PCDH10 will bind the virus, and they're thought to be, so garter snakes in particular, they're thought to be overwintering hosts. Okay, so in the wintertime, mosquitoes go somewhere, right? They don't keep flying around and biting people. So for a virus that is carried by mosquitoes, where does it go during the winter? And they may have a host, and here they think it's snakes. Because snakes, actually, I think they just get really lethargic during the winter.
Yeah, they get real slow. They slow down.
They hang out.
Yeah, they hang out.
So we don't actually know why these viruses use different receptors, and that's what this paper is going to address. There's a lot of structure analysis here and some virology. And the interesting part, the conclusion is that only 1 to 3 amino acid changes in the E2E1, the viral glycoprotein, explain the shifts in receptor recognition over the past century that I've just told you. Really cool. Okay, so the first structure they do is weave bound to PCDH10 from humans, all right, to get the baseline. This virus, we know it binds this receptor. So structurally, what does that look like? So they make virus-like particles, so there's no nucleic acid in it. They use a lineage C strain, which was isolated in Argentina, and this is being used actually in investigational vaccines. The soluble EC1 of the receptor, right, the terminal, the farthest from the membrane, that first domain, that binds the virus-like particles, okay, so confirming the interaction there. So then they do cryo-EM structures of the VLPs alone or bound to human PCDH, the EC1 domain only, not the rest of the protein, because the rest of the protein doesn't bind the virus, so you don't need to have it there. And you can see many, many features. I'm not going to go into them all, but for example, the protocadherin EC1 inserts into clefts on the E2E1 heterodimers, makes extensive contacts with E2 and E1 on both sides of the cleft. And from these structures, you can tell which E2, E1 residues, which amino acids bind, contact PC protocadherin H10. And you can see, for example, that the weave residues that contact protocadherin 10 are not conserved in EEEV or VEEV, which do not bind protocadherin H10. So right away you have an explanation for the different receptor bindings of these weaves and VEEVs and EVEs. Okay?
And there's numerous contacts, but you can pin the shifts down to just a couple of amino acids.
That's right. Yes, there are lots of contacts, and only a few are going to matter, interestingly.
Right.
Interestingly, they do some more studies of monomers and dimers. So E2, E1 can be monomers or dimers. They find that this EC1 of protocadherin that is contacted by weave E2, E1 is occluded in the homodimer, and only monomeric PCDH10 facilitates entry of the virus. So the monomeric one is the one that has to bind to. Okay, 14 human protocadherin EC1 residues contact the E2E1 of this virus that they've got the structure of. So that's the receptor residues. So these are highly conserved among human, marine, equine, avian, and reptilian protocadherin EC1s that serve as weave receptors. And so they want to know which ones are important, which of the interactions are important, which of these residues in EC1 of protocadherin are important for infection. So they're basically going to do infectivity assays. They use a cell line that does not have PCDH10 in it, and then they introduce modified versions of PCDH10, and then they affect and see if they can support infection. So they wanted to look at the North American strain. So they have chosen a bunch of North American lineages for these studies. And what they do is they change all the amino acids in protocadherin E1 that contact the E1, E2. And they say they substitute the amino acids with alanine. It's called alanine scanning because alanine is a small neutral amino acid that is not going to substitute for most other amino acids. So they find that changing most of the residues at the interface don't have any effect on entry of virus-like particles into cells, except for two, N40 and R42, that make polar contacts with E2. And when you combine these two changes, they abolish infection, right? So they abolish infection of the VLP particles from this this one of the North American strains. Now, we're not talking about the same one that the cryo-EM was done for. So you can identify the important residues, basically. I mean, these are looked at by homology from the cryo-EM structure, and you can identify important residues that are important for other lineages as well. All right, now, Imperial 181. Remember, this was isolated from California from mosquitoes. This binds protocadherin from sparrows, but not humans. All right, they want to know why. So they do the cryo-EM of Imperial 181. What a funny name, right? Imperial 181.
So it's just from Imperial Valley or something like that.
Yeah, maybe. They do cryo-EM of Imperial 181 bound to the EC1 of protocadherin. Okay. They can find all the contact residues and so forth and figure out why human doesn't work, basically. There's an amino acid L149 in E2 of the glycoprotein. It's highly conserved in weaves from lineage A, B1, and B2, and is replaced by glutamine in lineage B3, right, which is the most recent lineage isolated from the U.S., including Imperial 181. And substitutions there are thought to disrupt key hydrophobic contacts. And in fact, if they make that change, the virus cannot infect cells that make human protocadherin. So they say this L149Q polymorphism explains why some B3 strains don't bind human. So basically what's happening here, the enzootic reservoir is sparrows. And in sparrows, changes happen, and some of those changes randomly happen, and all of the changes randomly happen. And then when it gets into humans, some of those changes abolish binding to protocadherin because the humans don't matter for the biology of this virus, right? All that matters are sparrows. You got that?
Yeah, but I'm looking at this here. So the E2 variant that infects humans, that also that also infects sparrows. Is that correct?
Which variant?
I'm talking about the L149. I may be jumping ahead here a little bit. So
the L149 is replaced by glutamine in Imperial, right?
Right.
Which does not attach to human protocadherin, and that's the reason why it doesn't attach to it, right?
But my question is, does the L149, nevertheless, it must bind the sparrow PCDH10?
Yeah, it does, right?
It does. It has to. Yep. Otherwise you wouldn't be able to amplify the sucker. Okay, good.
Right.
All right. But occasionally,
in fact,
so that's that's a sort of a base strain. But if you mutate that E2, the receptor binding receptor binding protein in the virus at that position to Q, you can still infect the sparrows, but can't infect the humans anymore. Okay,
Exactly.
that's the imperial strain. Okay, good. Got
Exactly.
it. Got it.
Okay, imperial binds sparrow. It does have this L149Q change, right, which makes it not bind humans. This change disrupts contacts. So they're saying, well, the sparrow ortholog of protocadherin must make other compensatory changes, right? And they noticed some substitutions in the sparrow, EC1 of protocadherin. And they make a chimeric protocadherin where the sparrow EC1 replaces the human EC1 and the rest is human, right?
Right.
That allows virus entry into cells. So EC1 alone explains why imperial does not bind human protocadherin 10.
Okay, good.
So there are 6 residues that are polymorphic in EC1 in this region. Only 3, they say, involve residues that are close enough to directly influence receptor binding. And in fact, they show by mutagenesis that they can change human protocadherin 10. They can change those amino acids in the glycoprotein and make imperial bind to human protocadherin now, right? So basically the idea is that multiple interactions formed by sparrow protocadherin are not present in human, and you need those for imperial recognition.
I'm thinking about evolutionary pressures all the time when I'm looking at this.
Yeah.
And mosquitoes aside, we'll come back to that. What the virus needs, based on our preliminary discussion, is to replicate in birds. So it's not going to, or to amplify in birds, it's not going to, there's pressure to maintain that receptor binding specificity. And if you can do that a number of different ways, that's fine. If you can do that in a fashion that'll also bind human receptors, that's fine. But if you lose the capacity to, since the humans are a dead-end host, if you lose the capacity to bind to human receptors, that's okay.
Yep.
Right?
Yes, exactly. Because humans don't matter.
Right.
And neither do horses. All that matters are sparrows, right?
So you got to maintain that. This is interesting.
Yeah. It's very interesting, yeah. You always have to remember what's the reservoir, basically.
Yeah.
So for poliovirus, say, and I guess smallpox virus, the reservoir is humans, right?
Yep.
And if you mess up receptor binding, it's the end of the virus.
That's right.
It cannot exist anywhere else. But any virus with multiple hosts that are dead end and have a reservoir, then you can change things. Yeah.
That was a critical factor in getting rid of smallpox, is that there was only the human host for that particular virus. So if you got rid of it from humans, you were done.
Yep. Okay, so remember, some of these viruses bind very low density lipoprotein receptor. Right, so that's a protein in us that's important for cholesterol transport. But it also is a receptor for some viruses. So this receptor has 8 cysteine-rich repeats, which are called LA, and each of them have a calcium ion in them, and they usually interact with basic residues on the natural ligands of these receptors and also viruses. The basic residues in the alphavirus E2 and E1 that bind these LA repeats are not conserved. So they're all different. So they map these, the parts of LA that interact with a weave called Macmillan. Okay, this was isolated from a human in Canada in 1941. So this is a human isolate. It reproduces in humans. And it binds to VLDLR, okay? And they do modifications of the VLDR and put them in cells in culture and ask if the viruses can infect them. So what they find is that Macmillan has distinct LA repeat preferences compared with other alphas like Eve and Simlicki forest virus and Sinbis virus. So they decided to do the cryo-EM structure of Weave, Macmillan bound to VLDLR, which has 8 LA repeats in it. So they can see how these two interact in great detail during these interactions. So the viral glycoproteins interacting with the VLDLR LA repeats. They can see the residues that are involved, and I'm not going to tell you that because it's not really important for your understanding. But then they want to know what residues are important which contact residues are important. So they, again, alter the Macmillan particles, the glycoproteins, and they find that simultaneous LA repeat engagement of sites 1 and 2 on the Macmillan E1, E2 are required for efficient binding. So then they ask what naturally occurring polymorphisms, in other words, amino acid changes that you find in isolates on E1, E2 in these 2 sites on Macmillan might explain why lineage B strains don't bind VLDLR or ApoER2. And so they introduce these substitutions and then put the viruses into cells, and they can see that these 2 changes in the Macmillan abolish infections of cells that make VLDLR. So these 2 amino acids are required for VLDLR binding by Macmillan. So the point of all this is that you can use these structures now to figure out what are the contact residues, and then which of those are important for whether a virus binds or does not bind a receptor.
And in my mind, this all goes into a giant database that down the road is probably managed by artificial intelligence that tells us more globally about virus attachment protein receptor interactions, and ultimately allows us, and this is out almost in the fantasy realm, but I think the ultimate goal is, can we anticipate what's going to change the host range of a virus, okay, just by looking at sequence? And is there something on the horizon that we ought to be concerned about because this virus is tiptoeing into or near the human population?
Right. So would it be easy for the viruses circulating in North America, which are not infecting people anymore, to change so that they start reinfecting people again? And you see in the end, well, I already told you in the beginning, a couple of amino acid changes are all you need.
Right.
All right, so next they look at neurotropism. Many of these viruses infect neural cells. PCDH10 protocadherin and LDL-related proteins are on brain cells. Okay, Imperial 181, remember, isolated from a mosquito, doesn't infect people. Doesn't bind VLDLR, doesn't bind ApoER2, doesn't bind protocadherin H10. Replicates poorly in the brain of infected mice, so probably can't infect neurons. And so they want to know whether the inability of this virus to bind these receptors is why it poorly infects neurons. So they make changes in E2, which they can see are going to impact receptor binding, and this should restore binding to the receptors. And they check that, and they find that, in fact, the changes they make do restore binding. And then they take these mutated viruses and infect primary embryonic murine cortical neurons, okay, neurocells. So wild-type Imperial 181 doesn't infect them. We already told you that. And the mutants they made can infect the neuron. So when they restore, and we're not talking about a lot of amino acid changes here.
Right.
Couple, 2, 3, as we used to say, couple, 2, 3, is all it takes now to restore neurotropism with these viruses.
So, uh, 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 again, a bit of a broader context. We've isolated this virus called imperial from
Right.
birds that no longer infects humans. And without knowing anything else, maybe we sequence it. We see that there are a couple of changes in the virus attachment protein and the surface glycoprotein. And the presumption is that maybe that has to do with this change in host range, that it can't infect humans. Maybe that even extends to neurons, couldn't
Mm-hmm.
infect neural tissue. But that assumption is not necessarily true. There could
Right.
be all sorts of reasons that the imperial virus, just outside of receptor binding, might not infect humans or human neurons. This is the experiment that says all you got to do is change a few amino acids
That's right.
in the glycoprotein, and you've restored infectivity for humans. So this says that this receptor binding in this particular scenario for this virus is a critical host range factor.
Right. Exactly. So then they say, all right, now we know a lot about the interaction of some of these viruses with receptors. Could we use this, and knowing what the contact residues are, to predict a virus in terms of what it's going to do? And so they look at the South American weave strains, which some of which are not on any particular lineage. They look at lineage C strains, and they have L149 and E2, and they say we would predict that these bind to human protocadherin 10. Nobody's tested that. They don't have the right position in E2 to bind VLDR or ApoE-R2, and they confirm these predictions using infectivity assays. Okay, so that's they set out to do. And then they look at VLDLR binding strains in North American weaves that have been previously tested, and in fact, they show that E2K181, a single amino acid, is a determinant of VLDLR and ApoE-R2 binding. And this change has been acquired by strains outside of the old A lineage. So as the lineage is replaced, the other ones can acquire these changes to bind these other receptors. They next look at a strain called AG80-646. It is a weave strain isolated from mosquitoes in Argentina, and looks like it's really different from lineage C, right, which is the recently emerging lineage in South America. Now, all the weaves examined to date combine an avian receptor called MXR8. They found that this AG80 could not recognize sparrow MXR8 to infect cells. It has no affinity for MXR8, and so they think this represents another shift, right, that has undergone shifts in receptor binding properties. Now, so that implies that there's another reservoir besides sparrows for this virus, because it cannot infect sparrows because of these changes in the receptor binding, the E2 and E1 of the receptor binding part. Another interesting virus they look at is called Highland's virus. Highland's J virus is a North American alphavirus. It's closely related to weave. It circulates on the east coast of the U.S. It has an avian reservoir, and it's pathogenic in certain bird species. It is not considered to be a horse or human pathogen, and we don't know what the receptors are. But they look at the sequence of the E1, E2, and they know from their structures what residues are going to be important. And they use AlphaFold modeling and their data, and they say, if we made these changes, I bet it could infect horse PCDH10, and they show that it does. So again, you can make predictions. In fact, AlphaFold, you can take the sequence of a glycoprotein that hasn't been solved and make a predicted structure, see where the important residues lie, and change them. So they can say this is not too far away from being able to infect horses.
Very
Pretty
interesting.
Cool.
Yep. And all pinned on this receptor, glycoprotein receptor interaction, though, out in general, there can be other factors involved in the host range. But in this particular case, there seems to be a lot of it that's pinned on this receptor. This is very interesting.
Yeah. Now, Macmillan, remember, combined human and sparrow, protocadherin, human VLDR and ApoER2, sparrow MXRA8. So they want to know why. And it turns out that this they can look at the residues that control this. And it suggests that the E2 is more tolerant of individual substitutions when recognizing the avian protocadherin, but less tolerant with human protocadherin and VLDR. From their structures, they can see that there's a cleft between E2 and E1, and that's the major site of receptor engagement. That's where E1 of a protocadherin, for example, fits in. And so they look at a couple of related alphaviruses, VIV, EVE, Simliki forest, and WEAVE all bind the LA repeats of VLDR. The binding mode's different, which is based on amino acids that interact with this cleft, essentially. One last experiment. They make a VLDLR FC fusion protein, and they ask, could it block infection? So if you add this to virus and try and infect cells in culture, it blocks infection. And if you give it to mice, you give mice a single dose of this protein, 80% of mice survive infection. You give them 1,000 PFU, 80% of them survive infection. The control mice will die. So this, in fact, this works against EVE and WEAVE. These are viruses that can bind LDLR-related proteins. So a potential therapeutic approach, I guess.
So this is this thing, this sort of phony soluble receptor, is acting as a decoy, basically.
That's right.
And blocking entry, blocking attachment to the biological receptor on cells in the animal. If it were a therapy, it would have to be something where it would have to be applied immediately. Like, I know you've been exposed.
Yeah, that's
We're going
right.
to try and keep you from being infected. Yeah. Because they do this, one of these in the mice, they administer this decoy 6 hours before they infect. And they didn't do any sort of time series or anything like that. But my guess is if you let the infection establish itself, you're not going to be able to use this as a therapy. But it doesn't mean it's useless.
Yes, exactly.
Cool.
So basically we have here, they look at the polymorphisms, and WEAVE in particular, that have driven the shifts in receptor binding over the past 100 years. And they can use these. They can say, oh, these residues, we have to do surveillance to look for changes at these positions. For example, they say the E2K81 polymorphism, which if you make the right change there, can now bind VLDLR. You should monitor for that in environmental surveillance, right? And there are a couple of others. So basically they say, they conclude our findings provide a guide for estimating the potential threat any given WEAVE strain might pose based on sequence polymorphisms in its E2, E1 as determinants of receptor dependencies. So we know, as Rich said, the receptor interaction is key. Apparently, beyond the receptor, many species are permissive for infection, but it's the receptor that is limiting it.
So as I said, throughout reading this, I have the mosquitoes on my mind, okay? Because I'm thinking about the evolution of these things. I mean, why it's interesting that I'm going to get the names mixed up here. It's interesting that let me see, when did the imperial strain show up? Is that?
Imperial is from mosquitoes.
Yeah, and that is a newer strain.
Imperial
that has lost that has lost the ability to interact with human receptors.
Yes, I think 2005.
At any rate, as I'm looking at the evolution, I'm thinking, I wonder what the receptors are and what the various restriction factors are in the mosquito, right? Because they're going to influence all this. They're going to influence what is possible. They apply another selective pressure. So I spent a little bit of time trying to look up what was known about the receptors in mosquitoes and came up with not much. Okay. I think there's a lot there that is not known about the replication of mosquitoes. I'm not even sure the receptors have been identified.
Yeah, they may not have been. All right.
Cool.
All right, next, something completely different. Also a cell paper.
Oh, this is a great story.
This is one of my favorite stories.
Yeah.
Evolution of KoRV-A transcriptional silencing in wild koalas. Koala retrovirus. That's what KoRV, K-O-R-V, stands for.
And we have to start out here by acknowledging that koalas are like terminally cute.
They're very cute, and they're only naturally present in Australia. Okay, many places get them, like for zoos, but they don't exist anywhere else in the wild except in Australia.
And they exist on eucalyptus leaves, right?
That's right.
That are toxic to many other creatures.
So this is a paper from, let's see, the first author is Shan Xiong Yu, and the last author is William Thurkoff, University of Massachusetts Medical School, University of Queensland, which is in Brisbane, Australia, and Tongji University in Shanghai. And on this paper is my old friend Jeremy Luban.
Yeah.
You know Jeremy?
Yeah, I've met Jeremy before. He's been on TWiV, has he not?
Oh, yeah, he's been on a couple of times.
I seem to recall back in the old days, a couple of Jeremy Luban episodes.
Yeah, he's been on TWiV. All right, so as you know, retroviruses have to integrate their genome into the host cell. It's an essential step. So the virus particles have RNA, and they include an enzyme in the particle, reverse transcriptase, that copies the RNA into DNA, then it integrates into the host cell. And about 8% of our genome, human genome, consists of integrated retroviral DNAs of various kinds. None of them are infectious, but many of them are expressed, and some of them make things that we use, like the protein called syncytin, which is essential for placental development, and other things as well.
And so the next step in the replication cycle, once you're integrated, is to transcribe that integrated genome into either things that wind up as messenger RNAs after splicing, or unspliced stuff that is genomic RNA, and that can only happen from the integrated genome, which is why it is that the integration is essential.
So when the retrovirus genome is integrated into germ cells, that's called endogenization. And that's how the retroviral sequences are passed to offspring, right? You have to get into germ cells. If it gets into a skin cell or a lymphocyte, it's not going to be passed on because they're not germ cells, right? So it's called endogenization, and we know that this has played a big role in evolution of many species, as I've just suggested, but we don't really understand how endogenization occurs. Now, the KoRV-A, Koala retrovirus subtype A, it's a retrovirus that infects both somatic and germline cells of wild koalas. It's thought that they first acquired it from rodents many years ago, over 100 years ago. Some museum pelts of koalas over 100 years old have the DNA in them.
I remember this was an ASV talk.
Yes,
by some woman, I forget who it was, but man, it was an amazing talk. It had to do with, among other things, of collecting samples from pelts in museums, or taxidermed koalas, all right, and sequencing their genomes. Unbelievable.
So the koalas got it from rodents, and now it's spreading through koalas. And the KoRV-A, it infects somatic and germline cells, so it can be passed horizontally by contact to koala, but it can also be passed vertically from parent to offspring, right? Now, interestingly, the presence in the germline of KoRV-A is highest in the northern regions of Australia and lowest in the south. So this is called the north to south sweep. So basically, the transmission probably occurred initially in the north, and now it's sweeping through the population to the south. So this fact alone gives you an opportunity to look at endogenization and see how the host responds, right? Because the ones in the north are all endogenized and the ones in the south are not all endogenized, right? So that's the question that we want to address here.
And by the way, this thing in the koalas, when they, catch it as a horizontally transmitted disease, causes kids koala immunodeficiency syndrome.
That's right.
Which is, they're sick. And it's an AIDS-like immunodeficiency thing. And it's trashing the koalas, right? That's not good.
Exactly. It's very worrisome, in fact, that the koalas may go extinct. Yeah.
Right.
All right. So another part of the story are small RNAs called PWIs, P-I-W-I. So PWI is are proteins in cells, and the RNAs that interact with them are called piRNAs. These are 23 to 32 bases long. They are expressed in the germline, and they're thought to repress transposable elements. Transposable elements are sequences in our DNA that move around, right? They jump from place to place, and that includes endogenous retroviruses. They can move around. And these PWIs are thought to be defenses against them moving around in our germ cells.
Yeah, because if you think about it, you got transposons moving around all the time and reinserting in places. That's not necessarily good. That's going to cause damage. So there are these clusters of these PWIs, right, that repress this. This smells a little like, what's the system in bacteria? I'm blanking.
CRISPR?
Yeah.
A little bit, yeah.
A little bit, where you have specific sequences that serve as an innate antiviral defense.
Right. Now, their PWIs can be either antisense or sense, and they have different functions. So just think of plus and minus RNA or plus and minus DNA, one strand or the other. It can be antisense or sense. Antisense PWIs or piRNAs can transcriptionally silence DNAs. In other words, it repress the making of mRNA. And they do this by causing modifications of the chromatin. Those are the proteins that the DNA is wrapped around so that the transcription is shut off. Okay.
So these RNAs then must target chromatin modification enzymes to the relevant genes to do that modification. I had to think about that one.
Yeah, that's very cool. The sense piRNAs cleave RNA that's complementary to them, and that makes more antisense piRNAs, right? And so you're amplifying the amount of antisense RNA, and you get more chromatin silencing and so forth. There are these regions of DNA called piRNA clusters, and some people think when a DNA sequence goes into that, transposes into it, that causes silencing of it. And so it's kind of an adaptive genome immunity, if you will. And so how that happens, we don't know, but the analysis here in this paper of these KoRV-A retroviruses show that unspliced retroviral transcripts are processed into sense piRNAs, but spliced retroviral transcripts that include like the envelope evade processing and it looks like a normal cellular gene. So they're going to try and understand what's going on there.
So once again, the high-level view of this, correct me if I've got this wrong, but not only are koalas cute, and this thing is trashing them, but if I'm not mistaken, so we have a lot of ideas about retroviruses and endogenization and retrovirus evolution, but this is, if I'm not mistaken, the only example we've ever happened where we can actually see it happening in real time.
That's right. That's exactly right.
Okay.
It's happening in real time. Yeah.
So it's unfortunate for the koalas, but for us, we get to see this actually happening. Not only that, but if we can understand how it's happening, we may be able to help out the koalas.
Okay, so this is the basis for this study, and they're going to look at the genome, the transcriptome, and piRNA profiles of 8 koalas, okay, in the area surrounding Brisbane.
That the important thing there is that they can get tissues from those animals so that they can look at RNA. They do have genome sequences from hundreds, if not thousands, of koalas in the bank, where they can look at whether or not there is a provirus there, and et cetera. But it's only from the recently died animals that they can look at something close to an active infection. And they point out here, obviously, you're not going to go out and kill koalas to do this. So their sampling is very limited to koalas that have died in the wild for one reason or another, and they got to get to them fast enough so that they can take fresh samples and do this analysis. So that's a limitation on the study, but one they'll deal with.
Okay. So they have samples from koalas, testes, ovary, liver, and brain tissues from 3 koalas on the Sunshine Coast and 5 from Kurrumbin. So these are north and south of the Brisbane River.
Yeah.
And that's going to be important.
So cool.
Right? And they sequence their genomes and transcriptomes. So they find, this is very cool, between 73 to 89 and 72 to 109 KoRV-A proviruses in the germline genomes of the Sunshine Coast koalas and the Kurrumbin koalas, right?
So they both got a load of proviruses.
They got a lot of proviruses, and most are unique to each koala.
Point. Yeah.
Right?
If you look at proviruses in humans, you find a lot of conservation, right, of proviruses from human to human. But the fact that these are unique suggests that they're recent events, right?
That's right.
Okay.
Only one provirus is shared by all 8 animals from the Sunshine Coast and Kurrumbin, so on either side of the Brisbane River.
So an individual animal has dozens of these, right? So it's like you get infected with a retrovirus, you get the reverse transcription, and it just sprays these things all over the genome.
Right. And they move around. They duplicate themselves as well.
They move around.
Right?
Unbelievable.
So they look at some other, so koalas have other endogenous retroviruses from ancient infections, and they are shared among these koalas. So they're older. Right? And so these are newer, as Rich said. And what's interesting is that the KoRV-A often recombines with another endogenous retrovirus in the genome, and they can see that. But it doesn't seem to be functional. It doesn't replicate. Okay. So the Sunshine Coast and Kurrumbin are 200 kilometers apart on either side of the Brisbane River. But when you do RNA-seq of testes, KoRV-A expression is over 10-fold lower in the Sunshine Coast koalas compared to the Kurrumbin, right? The Sunshine is north, Kurrumbin is south. So north, 10-fold lower RNA produced from the retroviruses. And in addition, the animals north of Brisbane these are the Sunshine Coast, have fewer KoRV-A insertions than animals south. Okay? They're all endogenized, but they have fewer compared to south. Okay, so then they want to look at piRNAs, because we know piRNAs are known to silence endogenous retroviruses in the germline, as we just discussed. So they looked at piRNA expression in animals north and south of the river, right? So I love this study because there's wild animals, right? They're not inbred mice in the lab. They're wild animals that's natural, and we're looking at what's happening.
You've been to Australia. Have you ever seen the Brisbane River?
I have not, no.
I'm looking at pictures of it here because I was wondering, it must be a significant natural barrier. And it's quite a river. Very cool. Man, I need to do this. I need to go to Australia.
So they find that both sense and antisense piRNAs that target the koala retrovirus are equally abundant in koalas from north and south of the river. This is for established endogenous retroviruses, not the ones we're talking about. But in contrast, KoRV-A piRNAs are sense strand biased in the south, where in the north, both sense and antisense piRNAs are abundant, equally abundant. So there's some difference in the piRNAs in the north and south koalas as well. Cool?
Cool.
So KoRV-A piRNAs are sense strand based in Kooramin, and the other south of in the other side of the river, they're balanced. And they somehow feel that north-south river animals, the difference in the piRNAs, they think it's consistent with a transition in the function of these TWiV piRNA complexes that can do transcriptional silencing. And we'll talk about that now. Okay, so antisense piRNAs, remember, they guide TWiV proteins to genomes and do epigenetic repression, right? That's what you had surmised, Rich, right?
Yeah.
They guide them, say, methylating enzymes. So they look at methylation of the genomes. They want to see if this is correct and if there's some north to south gradient as well. So they have DNA from a sunshine and DNA from a Kurumbin koala. And in the both of these, most of the promoters of proteins coding regions that are known to be expressed are unmethylated, as you would expect, and the silent protein coding genes are methylated. All right, so that's a control that the experiments is working. And they say, by contrast, almost all of the KoRV-A promoters in the Sunshine Coast koala are highly methylated. Sunshine Coast is north of the river, right? Well, south of the river, the methylation is much lower. Okay, they have similar KoRV-A insertions, same place in the genome, but somehow the Sunshine Coast koalas in the north have gotten the ability to silence proviruses that are spread throughout the genome. Right, they're not just in one place, but they're silent, and above the river they're silent, and below the river they're not. Okay,
all consistent, all making sense. Of more really retrovirus, active retrovirus replication in the south, silenced in the north.
Now, normally you would say, remember, the idea about these piRNAs is that when a mobile element goes into a piRNA cluster, it triggers silencing. But they don't find any evidence that these KoRV proviruses are present in these piRNA clusters in the genome, right? So they're thinking, well, maybe a provirus, a KoRV-A provirus outside a cluster could somehow trigger silencing. And that trigger should be shared in the sunshine males and absent in the Kurumbian males, right? North, south. So they analyzed the distribution of all the proviruses in koalas using the DNA sequencing data. They find a provirus that maps downstream of a gene called MAP4K4. It's present in 27 of 83 sequenced koalas north of the river and not present in any of the koalas south of the river. The integration next to this gene. Okay? Amazing. North there, south not there.
And we're talking
it's
about the same integration in all 27 of these animals.
Same integration. And in some animals, it's homozygous, in fact, they say.
And so this is looking like something that's inherited, right?
They say it's probably positively selected, right? So integration of the KoRV-A provirus next to MAP K4K4 is overrepresented in the north and compared to the south. That's got to be positive selection. So there's some benefit to having it, and they go through some analysis of this and look and consider the geography. And what they conclude is that this MAP 4K4 provirus next to each other originated in an animal near Brisbane and is spreading through the northern population. And the Brisbane is a barrier, so it can't get across it.
Yeah, the river's a barrier.
And so this provirus integration must be suppressing KoRV-A replication because the animals north of the river, which have mostly this, don't have lower copy number of the KoRV-A compared to animals south of the river, right? So they want to know what's going on here. What's the link between this integration and silencing of KoRV-A? And they find that KoRV-A is expressed 9-fold lower level in heterozygous animals relative to animals that don't have the provirus. This is expression of the genome, consistent with positive selection of this driven by silencing of KoRV-A. So KoRV-A is likely detrimental. And if you have this integration near MAP K4K4, it silences the provirus. And it seems to do so in a genome-wide fashion, not just that one provirus, right? So that's beneficial, and that's probably why it's positively selected. But they did find 2 animals just south of the river that had this integration, and they silenced all the proviruses. So they call the river a soft barrier to migration.
It'll make it down there eventually.
Yeah, animals have been able to get through there a little bit, and eventually it'll get down south as well, right? So basically, these antisense piRNAs, they guide, remember, they do transcriptional silencing, and the KoRV-A provirus is downstream of this MAPK4 gene, and they think maybe there's read-through transcription from the gene to the provirus, and that generates precursors for piRNAs, which then silence, right? The piRNAs can go anywhere.
So I don't know if you said this specifically, and in the paper they sort of waited to lay this one on you, but the proviral integration into that gene next to that promoter is in an antisense orientation, right?
Right.
So that's all set up to make antisense piRNAs.
Right.
Jeez.
Right. So they do some analysis of the contribution of this provirus to the KoRV-A piRNA pool, and they basically conclude that piRNAs derived from this provirus are loaded into a nuclear PWE protein, and these complexes guide silencing of proviruses throughout the genome from dispersed elements, they call them. So that's the defense of the germ cell against transcriptional elements. In this case, integrated next to a gene. Read-through transcripts make antisense piRNAs that then transcriptionally silence all the other and why is it specific? Because the piRNA is derived from the KoRV-A genome, right?
So this is all of this retrovirus evolution and innate immunity in real time. So
it's amazing. It's very cool.
If you inherit this particular provirus, you're immune to the disease.
Right.
And I wonder how long it's going to take. I wonder how long we have to wait to resurvey these animals and look and see it spread south. And
so
I don't think this soft barrier is critical to this whole thing because the southern group is the control group in a way, and the northern group is the experimental group, or the other way around, however you want to look at it. But if you just had a population where you didn't have that soft barrier and you couldn't see the difference, it'd be a lot more difficult to interpret.
Yeah. So basically, this barrier has let them figure out what's going on because there's a difference between the animals north and south of the barrier. So the animals north just randomly happened to integrate next to this gene, and that helped. So that was a positive selection. So more animals were born that have that, and they didn't get there yet south of the border because the animals from the north didn't cross it, but eventually it will, right? That's the idea.
Yep.
The animals in the south, they haven't been able to silence their KoRV-A proviruses, so they're a little bit more screwed, right? Actually, there was a part here that I missed because the president they speculate that these transcripts are what's the word I'm looking for? They hurt the animals. And so there's positive selection to have a mechanism that gets rid of them. Adaptive silencing, this is called, right?
You know, everything is Australia is too damn big, and everything is really far from it. You know, if you're going to visit Australia, you need a lot of time. Brisbane's a long way from Sydney, is a long way from Melbourne.
Yeah.
Melbourne is a long way from Adelaide, is a long way from Perth. Jeez.
So they say that basically this MAPK4 provirus which established in the north, is sweeping through the north by positive selection because it's beneficial, because it's bad to have the KoRV-A expressed. And they say this probably is the earliest stage of a cluster formation, right? Because that's the idea that these transposons when they go into clusters, that's when you get immunity to them. And so maybe this is as a beginning of a cluster formation. So we can watch this as it goes. Maybe it's going to have other transposons come into that as well. So they say animals that don't have the map for provirus they make a lot of envelope, and here's what I wanted to say. This envelope, which is the viral spike protein, could compromise male fertility, leading to a reproductive advantage in animals that carry the MAP4K4 provirus and suppress envelope expression. So that's the reason why it's being positively selected. Very cool story. Very cool. I mean, I knew about this, but I didn't know the Pee-wee connection and the differences. This paper is the first one to show it between the North and the South. So they end up by citing Barbara McClintock, who discovered transposable elements in maize, right? She said genotoxic stress activates transposons, and said that this mobilization, moving around, could provide beneficial genetic diversity that contributes to adaptive evolution. And that's exactly what they have seen here. She was so smart, right? The provirus went next to this MAP4K4 locus, and now pRNAs are made that are suppressing transcription. Very cool. So jumping around transposition can help sometimes. Sometimes it hurts too. You like this story, Rich?
Oh, man. I'm just looking up Barbara McClintock because I want to get this right.
Nobel Prize, worked at Cold Spring Harbor for many years.
Cold Spring Harbor on maize, discovered transposable elements. She was born in 1902 and died in 92 at the age of 90, and just sort of poked away on her own. And for a while, people didn't take her too seriously.
That's right.
I think they didn't think this transposable element stuff was, DNA was supposed to be all stable and everything. But she just plugged away and proved it, and transposable elements are really important. And as you say, got a Nobel Prize. When was that? National Academy, 1945. National Medal of Science, 1970. Blah, blah, blah. Nobel Prize, 1983, at which point she would have been 81 years old. Took a while. And I'm looking at the koala distribution. Koalas exist on, the current range is a lot less than the historic range. But they're all up and down the east coast of Australia. And it looks like Brisbane is kind of right in the middle north-south wise of that whole thing. Very, very, very interesting. What a great story. And so educational. It's I don't know, it's nerdy in a way, but it's really important. I mean,
Well,
let's
yeah.
remind people that HIV is a retrovirus, right? And that when did HIV jump from chimps to humans? 100 years or so?
1920.
1920?
Yeah.
Okay. So all of this stuff has happened to humans, all right? And we're not necessarily done. This is all an active process. So
Yep.
this is really interesting. It's too bad for the koalas. I'm glad they've got this MAPK antisense insertion because there's hope. I wonder, do we have any? Well, we've got all sorts of antiretroviral innate immune defenses in humans. I don't know about specifically HIV. Well, I don't want to get into it.
Well, HIV doesn't do germline, so there's no TWiV defenses against it. Right?
Of course. Yeah.
So none of this would apply. Yeah.
Right. Okay.
All right. Let's do some email. Can you take that first one, Rich?
Sure. Hang on. Philippe writes, "Hello, TWiV. I have been following your post for the past couple of years. I find the work that you all do breaking all the misinformation is essential for building a stronger future. In regions where uncertainty and misinformation are rising, public trust in vaccines is being undermined, often perceived as a lack of transparency from manufacturers and regulatory agencies. While I understand the interest in using placebo-controlled trials to demonstrate efficacy, this approach may not effectively address the root concerns of vaccine-hesitant individuals. Their hesitation often stems not from doubts about effectiveness, but from fears of vaccine-induced adverse effects, particularly long-term or rare conditions that they feel lack adequate acknowledgement or redress. The core of the issue is the perceived lack of accountability. Because pharmaceutical companies and government agencies are protected from liability in most cases, people feel they have no recourse if they suffer side effects. This distrust is amplified by the emergence of long COVID and other post-viral syndromes that some believe are underreported or misunderstood. To rebuild trust, transparency around side effect and open acknowledgement of rare but possible outcomes are essential. A placebo study may demonstrate efficacy, but it won't address deeper concerns about safety and accountability. A more meaningful approach might include improved communication of risk data, greater public access to adverse event tracking, and policies that show commitment to patient safety and support. I think you'll find the following context relevant. Medical knowledge is now doubling every 18 years, while fields like nanotech double every 2 years. Meanwhile, drug R&D efficiency in the U.S. halves every 9 years, a trend known as Eroom's Law, the reverse of Moore's Law. If unchanged by 2043, developing a single drug could cost $16 billion. This economic pressure risks narrowing innovation to only the most profitable drugs and not the most needed ones. Regard, Philippe. All well said, Philippe. We do our best. I would, that's the unfortunate thing about the current trends is that we now have people who are serving in an official capacity who are fanning the flames of the conspiracy theories and the distrust of what in fact are reliable therapeutics and vaccines. It's a shame. So that makes the task of countering the misinformation, it more than doubles it. It makes it much more difficult.
And the other problem is that the people in charge now have the biggest megaphone and they're spewing the misinformation, right? everyone else is kind of small compared to them. So it's very hard. But I agree that just having a placebo study is not going to convince people because of lack of trust. So, that's, as you said, we're trying to help with that, but we have a limited audience. I appreciate, Felipe, your thoughts, though.
Yeah, absolutely. Absolutely.
Everyone has good ideas, so please send them in.
And there is, I think the news article that we did up top with FDA and CDC collaborating in a report that they were stopping this particular vaccine. Now, some are going to look at that and say, oh, they made this vaccine and it's terrible, it's messing people up. But really, the perspective ought to be, this is transparency, okay? And people do take this seriously, and they're not trying to hide any of this stuff. When there's a problem, it is brought to the surface. And there is no pharmaceutical company or federal agency that's trying to suppress this stuff. They're, in fact, publicizing it and making sure people know about it and taking appropriate action.
Jason writes, I cannot tell you how grateful I am for your podcast. I am just a simple layperson living out here in our crazy society. You guided me through the pandemic and allowed me to see truth from fiction. You continue to do so. Once during the podcast, one of your scientist guests mentioned they listened to The Atlantic. I'm always trying to find the most trustworthy sources of information. I figure if it's good enough for the scientists, then it's good enough for me. So I bought a membership to The Atlantic. I've been very pleased. Obviously, no source will be 100% truthful, even if they give a good faith effort. But there is a continuum of sources of truth as measured by their good faith, rigorous process, self-checking, looking at various sides, etc. I'm so happy your guest mentioned The Atlantic. In fact, last year, Brianne was praising it, and I got a subscription too. I really like it.
Yeah, I subscribe as well. It's a great magazine.
In this age of disinformation, I would love it if you and your guests could much more frequently mention the sources of truth you go to in non-virology domains. I feel like this would be better than knowing the weather and definitely more practical and impactful. Of course, all expertise is domain-specific, so your sources in non-virology arenas could be utterly off base, but I'm willing to bet that my life and the lives of your listeners would be better off if we heard which news sources and non-virology podcasts you all listen to. I really like Malcolm Gladwell.
Yeah, I don't actually listen to him regularly. Sorry, Malcolm. But I've listened to a couple of his podcasts. Yeah, he's very good. My go-to source, I've mentioned this many times, every night is the PBS NewsHour. Okay? They really do genuinely provide a balanced view of what's going on. They have both conservative and liberal individuals, mostly people who just think well and don't really necessarily have a partisan slant. And I would say, as long as I'm plugging them, that at least locally, now I don't know if this is PBS or NPR, National Public Radio, same deal. Okay, it's the same kind of organization. But they rely heavily on contributions, and they rely heavily on government funding, which is in jeopardy, as it always is. But now there's a potentially really serious challenge to government funding the public broadcasting system. At any rate, the subscription rate, there's less than 10% of listeners that subscribe to either NPR or PBS, and that to me is a shame because these organizations probably wouldn't need government funding if everybody who listened, in particular listened regularly, would pony up. And my guess is, I don't think, even if the government withdraws funding, I don't think these things are going away. They're too well established, and maybe people will pitch in. But this is really great information.
I mean, hopefully people will pitch in now that
hopefully
government funding has been withdrawn.
Have you seen there's a great, I ought to pick this sometime. There's a great thing from years ago when, public television has been under threat for government funding at several different times. It's always under threat, sometimes more intense than others. But I don't know, 20, 30 years ago or something like that, it was on the verge of being cut. And they had Fred Rogers, as in Mr. Rogers' Neighborhood, come and testify to Congress. And he gave testimony that gives you chills and wants to make you cry. And the senator interviewing him said, well, I think you just won $20 million.
Cool.
That's very good.
I didn't finish his letter. I'm a CPA, and I'm well aware of the plethora of bad advice and self-serving advice out there in regards to financial services. I'm just so tired of all the lies and falsehoods and advice that isn't fully in the interests of the listeners. Look at what it's doing to our society. Okay, well, thank you again for all you do. Obviously, feel free to ignore my request or do as you see fit. Jason, we think it's a good request, and I do think we mention non-virology sources often, and sometimes our listener picks are those. So just
and actually, my other go-to resource, I must access it at least a dozen times a day, is Wikipedia.
And Wikipedia is very good.
And remarkably, when I access it in areas where I actually know something, I can see very little inaccuracies. I think it's extraordinarily accurate. So that's one of my first sources of fact check. The other one that I picked a while ago is Snopes. When I see something and I'm, a piece of news or something that looks like it might be misinformation, one of the first things I do is Google Snopes, that's Snopes, and then some sort of keywords for what might be misinformation. And Snopes is a fact-checking resource.
Right.
And that quite often triggers a response. If Snopes has looked at it, that'll pop up, and that's very helpful.
Rich, you're next.
Patrick writes, Dear TWiV, you indicated the Integrated Research Facility, IRF, located at Fort Detrick is shut down over security concerns. It is important to point out that this is not the same facility as USAMRIID, where you conducted interviews with virologist John Dye recently. The IRF is run by the NIH, while USAMRIID is run by the Army. The shutdown will not likely pose any serious threat to our biosecurity or threat agent repositories you lamented. The NIH also has the Hamilton facility in Montana, as well as the Needle in Boston. In addition, facilities at UTMB and San Antonio are still up and running. We are not left without capability as a nation. While Jens Kuhn worked at IRF, he will always find a place to be productive as his skill transcends the boundaries of the RF. Sincerely, Patrick, who was previously a division chief at USAMRIID. Yeah, I appreciated that. As I recall correctly, there are 2 BSL-4s in Fort Detrick. Is that correct? USAMRIID and IRF? Is that correct? And I do recall being quite impressed when we visited USAMRIID in the role that they played in the COVID pandemic, because they were poised to do a lot of analysis that others weren't as well poised to do. So this is good. Do you have any news of that IRF shutdown, Vincent? Since it was announced?
Don't, not any more than this, no.
All right. Good. Thank you, Patrick.
Isla writes, I'm a molecular biology undergraduate student at the University of Sheffield in the UK, and I try to listen to TWiV and TWIM as much as I can. I found Sunday's episode incredibly eye-opening, and I just wanted to say, please do not stop talking politics. As someone not in the U.S., we hear headlines, but not how they are affecting science and scientists as individuals. I think the UK and many European countries are heading in similar political directions as the U.S., and so it is always good to be informed. In any case, I think it's great to have recordings of what is happening and at what time, as you never know what will be erased or hidden. So please don't apologize for bringing politics up. I really enjoyed hearing it in context, and the rest of the world is on your side. Thank you, Isla. That's great.
Yeah. This is what I discovered going to the meeting in Croatia. As the reaction was not generally, are you Americans completely crazy? The reaction was more like, I'm so sorry this is going on. All right. Because, nobody's immune to this nonsense. And it's going on in lots of different places. We all have to help each other out to get through it.
You're next.
Edward writes, Dear TWiV team, may I offer my heartfelt condolences to all colleagues and associates on the departure of your friend Dixon. Were the wider world as erudite as he, what a wonderful planet this could be. Warmest regards, Edward. Yeah, Dixon was quite the Renaissance man, and we do miss him. Thank you.
I mean, he's missed all of this stuff, right?
Yeah.
I just would love to hear him on about it.
Yeah.
Bob writes, Dear TWiV, early in this episode, you mentioned that one could be tested for the entire range of infectious agents causing respiratory infections by using BioFire Film Array Respiratory Panel. This is true. NAT are wonderful things. However, one cannot go to their primary care and get them to order this testing on you on demand. Your insurance will not pay for it in the outpatient setting. That test runs probably about $700-800 a pop. I'm a retired 522 medical technologist with a specialist registration in microbiology through ASCP. We ran that platform at Bronson Methodist Hospital in Kalamazoo, Michigan, a 425-bed metropolitan facility with a level 1 trauma center. We used it on the critically ill that were going to be admitted to help determine treatment and isolation. Thanks. I love both TWiV and TWIM. Robert's in Kalamazoo, Michigan. Thanks. That's great. Great to know. Yeah, I didn't think it was is something you could order.
Well, it's interesting because the reason that I'm familiar with it is that not too long ago, my grandson with a respiratory infection went to somewhere. I thought it was just a doc in a box that did accept whatever my son-in-law's insurance was.
Yeah.
And they ran what I presume is this panel or a very similar one on him. And I'm sure that my son-in-law didn't pay for that out of pocket. So
yeah,
I don't know what the story is.
Let's see here.
David?
We can finish these.
Oh, wow. Been a long time since we've done that.
You're next.
So David writes, Hi. We used to do all email episodes. That was when we really used to accumulate them. David writes, Hi. We now have an array of specific antiviral drugs. They all work best when given early during the phase of active viral replication. Multi-pathogen diagnostic panels make sense and would allow us to give the correct drug as early as possible, I suppose that is. The big impediment to making multi-pathogen panels widely and rapidly available are antiquated reimbursement policies. Insurance companies won't approve reimbursement for a multiviral panel unless a single pathogen test has already been run and was negative. Okay, so that's informative. By the time you go to the clinic, run a flu or a COVID test, get back the result, schedule a second visit, and return to the clinic for the multi-pathogen panel, wait for the multi-pathogen panel to read out, have the appropriate prescription sent to the pharmacy, go to the pharmacy to pick up the drug. The efficacy of the drug will be greatly reduced by the long delay. Multi-pathogen diagnostic panels were once regarded as fishing trips in the area of incidentalomas and less is more medicine. Their use was discouraged. Time to update the guidelines and recognize that speed to delivering appropriate therapy is critical, and patients can be infected with multiple pathogens. That's Hickham's dictum. Beth David. P.S. Oh, this is, I'm going to do this. This is David States, who I've worked with before, who's Chief Medical Scientific Officer of Angstrom Bio in Austin, Texas. I communicated with David during the pandemic about this very issue of diagnostics. P.S. On the Gates Foundation, there is a tax rule that nonprofits must pay out at least 5% of their net worth each year. This is to prevent an organization from forever accepting gifts and never spending the money. If Gates or another foundation makes more than 5% return on investment, it could be perpetual, but they do need to keep spending. And he gives a link to that. That's in reference to my, among other things, I think my comment that why not have the Gates Foundation create an endowment like the Howard Hughes to let it go indefinitely. And we discussed that. And this is some technical detail relative to that. Great. Thank you, David. Good stuff.
Anthony writes, it was the first Trump administration that lifted the Obama pause on gain-of-function research funding, not Biden. So he gives a link to that. Wow. I didn't know that. Then he writes, the science center of gravity was Germany. The Nazis changed that. That's a little scary, isn't it?
Yeah. We live in interesting times.
Go ahead for the next one.
Karen writes, hi, I've been listening to TWiV and especially the clinical updates since early 2020. I have learned so much that I regularly share with others. Thanks so much for continuing to provide these podcasts. I want to share a couple of items related to science misinformation and the NSF funding cuts. Our daughter, Molly Edwards, PhD, plant biologist, science communicator, and educational media producer, and gives a link to Molly's website and YouTube site, has been creating social media posts on the NSF funding cuts. Here are two examples, and she gives examples of those on Instagram and Blue Sky, and we can put those in the show notes. Also, I don't know if you have heard about the Cornell University Advocacy Project developed by undergrads in the Cornell Science and Policy Club, the McClintock Letters. It gives a link to that. This project is supporting and training scientists to write letters to the editor in local papers featuring the importance of federal funding for scientific research and etc. The letters are to be published on Barbara McClintock's birthday, June 16th. As you know, she won the Nobel Prize for Genetics Research. How ironic that we should do that in the same same episode as we just talked about Barbara. Leave it to the undergrads to come up with something this creative and innovative. Kudos to them. Thank you for all you do, and thanks for your emphasis on science misinformation and science communication. As a retired pediatrician/public health professional, I am incredulous about the misinformation that the administration is sharing concerning vaccines and health, and I'm so angry about the funding cuts that will affect the health and well-being of children and their families. It is shameful. The damage will take so long to repair if it is reparable. Thanks, Karen. Karen is both an MD and an MPH.
Yeah, thanks for the link to your daughter's work.
Mm-hmm.
Take a look at that. John writes, and this is the last one, asking for full testing of the new Novavax formulation is like asking for complete crash testing of a new car model that differs from last year's by a different set of hubcaps. Very good.
Great analogy.
Also, with respect to fluoridation, And close to home for you both, and Brianne, I saw a few months ago that only something like 16% of counties in New Jersey fluoridate their water. Astonishing. Yes, it's true. It gives a link to that. Overall, Pennsylvania doesn't score that well, but checking elsewhere, I've seen that here in Allegheny County, PA, 93% of water is fluoridated. Also noted, Montana, Minnesota is among the highest, compatible with how I generally viewed the state before you brought the mRNA bill to my attention. And Emigerd, Kentucky is even higher. Does that mean that Kentucky whiskey is most likely fluoridated? That's a good question. I bet that they use clean water. Yeah. Otherwise, Greater Braddock is experiencing lovely spring weather, full sun and hitting 74. Hope your results are the same.
So according to my news source, PBS NewsHour, yesterday or within the last couple of days, Florida banned fluoridation in its water. And I think Utah, DeSantis described it as imposed health care or something like that, So this is riffing.
It works and no side effects.
Yeah, this is riffing on the whole mask mandate or vaccination mandate outrage. Okay, so you can score political points by banning something that looks like it's imposed. Well, they will. They'll make an interesting control for the benefits of fluoridation in the long
run. Public health doesn't work unless you mandate things. That's the way it is. You have to give up those freedoms if you want to be healthy. By the way, you can buy fluoride tablets for states that don't have fluoridated water. And it's good not just for kids, but for adults, right, to keep your teeth healthy. I should check my county here, see what's going on. And if it's not there, I'll get some. I use fluoride toothpaste too, right?
So I'm reminded again of the two different maxims that we have discussed on this show before. It's worth repeating that public health, these came from two different individuals, but they line up with each other. Public health is by definition a mixture of science and politics. That's observation number one. Yeah. Observation number two, which I think came from Peter Daszak, is that if you mix science and politics, you get politics.
Yep, that's right. Okay, Rich, let's do some picks of the week. What do you have for us?
So this has been lying in and I hope this is accessible to everybody. I think this is saved as a shareable link to the Washington Post. This has been lying in my inbox, my TWiV pick box for nearly 2 years, and it is an MD's blog titled "What's That Stuff in Your Belly Button? Science Has the Answer." And just briefly, this is a short article about various scientific studies that have looked into what it is that collects in your belly button and under what circumstances. So this is really important stuff. It turns out that it's mostly lint, skin cells, the occasional bacterium that is of no particular consequence. And there are even studies that talk about the configuration of the belly button and how hairy it is around your belly button that influence what collects. There's an engineering study that talks about how the hair around your belly button actually harvests the lint from your shirt and collects it in the belly button. Okay? And it's one of these studies that looked at thousands of samples of this guy's, I don't know if he collected from his own belly button or multiple belly buttons, but it's published. Won an Ig Nobel Award in 2002. So for your amusement, in the midst of all of the other nonsense going on, here we go. If you wanted to know what's in your belly button and why, This is for you.
So what? It collects stuff because it's a depression, right?
Yeah. Yeah, if you got an outie, you don't collect stuff.
Yeah.
And different types of innies collect stuff at different rates. I don't know if they collect it. Yeah.
So interesting.
You need this.
That's cool. My pick is an article in Science Daily, which is basically a website that summarizes research papers, so in a simple way so you can understand, and then they give you the link to the paper. So this is called New Study Reveals Our Skin's Own Bacteria Can Help Protect Us from the Bad Effects of Sunlight. So the sun microbiome has been shown that it exists. It's bacteria on your skin. They have all kinds of interesting functions. Some of them make antiviral peptides, for example, that protect you. But now in this paper in the Journal of Investigative Dermatology, they show that specific skin bacteria protect you from UV. And they do so by metabolizing cis-urocanic acid using an enzyme called urocanase that allows the skin to adjust how it responds to UV radiation. So I think this is very cool.
Very cool.
Microbiome is just amazing.
Yep.
We did a paper on TWiV yesterday, which showed, so there's this disease called fibromyalgia, right? 2% of the population have severe pain with no obvious reason, right? There's a mouse model. Well, what you can do is you can transplant feces from people with fibromyalgia into mice, and the mice have higher levels of pain.
Oh, wow.
Really cool. So microbiome, man. So anyway, the skin, I don't know what you do with this. Spread bacteria on your skin or maybe something else, but it may lead to therapies. I think that's very cool. We have a listener pick from Rona. Dear Vincent, during the latest Q&A, that's a live stream I do Wednesday nights, I enjoyed your lapsing into French. When you were talking about Italian being the most beautiful language, I remembered that I wanted to share with you a film I saw in connection with a Holocaust Remembrance Day in April. My friend Vincent Marmorale, president of Italy and the Holocaust Foundation, made this film called My Italian Secret, The Forgotten Heroes. Did you know that many Italians helped save approximately 80% of the Jews in Italy during the Holocaust? In the film, we are introduced to Dr. Giovanni Borromeo, the director of a hospital in Rome at the time of the Holocaust. He invented a disease called K to deceive the Germans and save Jews. He hid the Jews in this hospital, in quotes, and told the Germans that entering could lead to contagion to this deadly incurable disease. Thus, the Germans decided not to enter the hospital. This is why I am picking Vincent Marmorale's film, My Italian Secret, The Forgotten Heroes, and his charity, Italy and the Holocaust Foundation, as my picks of the week.
Outstanding. That's great. I like that story.
I think I've heard this before.
That's very good.
About this hospital, yeah. Thank you, Rona. Really nice. That'll do it for TWiV 1219. You can find the show notes at microbe.tv/TWiV. You can send us questions, comments, picks of the week. I love getting your picks of the week. We all do. TWiV at microbe.tv. And if you like these programs, consider supporting us. Microbe.tv/contribute. Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Thanks. Always a good time. This is fun.
I think you and I have done solos before.
Oh, yeah. First of all, we've done a lot of them on the road.
Yes.
Okay, because I tell these stories all the time. We went through a period of time when we were both on study section, and we made it a point every time there was a study section meeting to spend an extra day or so there. And sometimes it was in D.C., sometimes it was on the West Coast, and pick out a few people to interview. That's when we, one of the, there were several highlights, but we interviewed Tony Fauci.
Yeah.
We interviewed Eugene Koonin, Bernie Moss.
Don't forget Eric in California. DNA is a real problem.
Eric Delwart. I was having trouble remembering his last name a while ago. And Michael Emmerman. Did we interview Harmeet Malik, or did we just meet him when we were there? I don't think we interviewed him.
No, we didn't interview him. No.
Okay, but that was a memorable trip to the Hutch. Yeah, we've done several of those, several of them remote. And then in the old days when there were fewer of us, it would happen like this occasionally, that there was just the two of us.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.